everyone. I'd like to thank the organizers for giving me the chance to talk to you today about our work with using machine learning to try to understand the dynamics of activity across the brain. So brain activity occurs at many different scales. We have things that are happening at the level of synapses, very tiny, organized into layers and columns, and then into these larger networks like, say, thalamocortical networks or sensory motor networks, and even widespread modulation of the activity of the whole brain by neuromodulatory systems, for example. And these also happen at different time scales. You know, things can be on the order of a millisecond for some of these really fast transmissions and action potentials. Or we might be talking about changes that occur over minutes or hours or even days or years if you're looking at how the larger scale activity of the brain might vary with changes like experience. So all of this stuff is mashed together into the resting state fMRI signal that we use to map functional connectivity. Now, we like fMRI because it does give you whole brain coverage, it's non-invasive, it has reasonable spatial and temporal resolution, even though it's not directly sensitive to neural activity. So we're kind of getting this integrated and filtered signal that tells us something about what's going on with the neural activity inside the brain. And the type of data we get is large by nature. So from any given subject, we have tens of thousands of voxels, maybe even 100,000 for high resolution studies, and often thousands of time points. Now, the way that we usually look at this data, and have over, say, the past 20 years or so, is to do something that I'm going to call time average functional connectivity. And that means that maybe we take the brain and you can do it on a voxel by voxel basis, or you can also take the brain and divide it up into different parcels, so areas that are kind of functionally similar and work together, so that we can reduce that spatial dimensionality of the data that we have. And then you can look at the relationship between the signal from each of those areas over time with all of the other areas. And you get something that looks like the correlation matrix that's shown on the right side of the screen. So each of the rows here is an area in a given network. Each of the columns is another area in a given network. And of course, the signals from the signal of an area in a network is highly correlated with itself, and so you see that along the diagonal. But you can also see these blocks of correlation where our parcels have been organized into networks that exhibit high correlation within the networks, and then you can also see interactions between different networks that you see in these blocks off the diagonal, where some networks work together and are correlated, and other networks kind of go in opposition to each other and are anti-correlated. So what's wrong with this? Nothing. It's a great way to summarize data, but we lose so much when we go to average measurements of functional connectivity. So we're taking, say, if we're talking about the Human Connectome Project data, we're taking 100,000 voxels, 6,000 time points per subject, 1,000 subjects, and we can condense all of that into one single matrix of 360 by 360 parcels that describes the average functional connectivity across all of these people. So that's a great start, but we're losing all of the information about time-varying activity. And we know that different things are happening in the brain over the course of the scan. You know, even if you're doing a task, your mind wanders away from the task. If we're looking at resting state studies, as we often do, you might be thinking about different things. You know, maybe you're thinking about where you want to go on vacation, and then all of a sudden you think, well, I've been in the scanner a long time. Isn't it time for lunch yet? Or you might be in there getting drowsy or progressively more uncomfortable over time. And all of these things can influence what we see in the brain's activity. We know that if we look at this time-varying interaction between areas that we measure with time-varying functional connectivity, we see relationships to behavior. And I've cited one paper here that was an early one from my own group, but there are many, many papers now showing that these time-varying interactions in the brain are relevant for behavior and for looking at how brain activity changes in neural disorders, for example. So we need to find ways that allow us to capture the dynamics of the whole brain as a complex system. The current approaches for time-varying analysis are mostly based on kind of variations of our average functional connectivity, with the very simplest being sliding window correlation, where instead of taking the activity and looking at relationships like correlation that persists for the entire length of the scan, we chop that scan into segments, maybe 30-second segments, maybe 60-second segments, and we calculate correlation for each of those segments. And then we can move that segment along or that window along and see how correlation between those areas varies over time. Another option that I'm going to talk about more later is looking for repeated spatiotemporal patterns that we call quasi-periodic patterns, or QPPs. And these are basically detected using an autoregressive algorithm that goes in and looks for things that happen over and over during the course of a scan. Another common approach is to look at coactivation patterns or individual events. In this case, you're looking for times when the activity in a certain area or group of areas passes a threshold and trying to average those together and see what else activates with those particular areas. And all of these approaches are often followed by clustering to further reduce the data and make it more interpretable. So these have had a lot of success. We've seen a lot of differences within and between patient groups, for example. But I think that they all assume particular models of what's going on within the brain. And I think we still have room to improve and try to create better models to help us better analyze the spatiotemporal dynamics of the brain from this complex systems perspective. So one of the things that we tried is to just use dimensionality reduction on resting state brain activity to try to map it to certain states. We did this partly because we've done simulations that show looking at the raw data is usually closer to the underlying signal than looking at sliding window correlation. And so what we did was assume that each scan is one time point in a highly dimensional space. And then we do this t-distributed stochastic neighbor embedding, t-SNE algorithm, which basically takes this high dimensionality space and maps it onto a low dimensionality space, 2D here, using gradient descent. And I've got a picture of a fruit fly over on the side because we were inspired to do this by Gordon Berman at Emory University, who uses the same technique to look at classifying behavior in fruit flies. So we like to think of this as describing brain behavior. So what we actually did is to take all of the HCP data, including the task scans, the rest scans. We had 446 volunteers from the data set we used, and each of those had 8,680 time points. We took that data to reduce the spatial dimensionality. We ran independent component analysis, and we took 50 components from that ICA. At the same time, we found, through basically trial and error, that we needed to expand the frequency representation of these signals. And so we applied the continuous wavelet transform to those 50 ICA components to kind of tell us more about what frequency was doing what at what time. And that's the data that we threw into the t-SNE algorithm. So on this slide, at the top left, I'm showing the latent space that we got as the output of t-SNE for the rest scans and the task scans, shown separately in A and B. And what we thought was really interesting is that there's not a lot of overlap. Yes, I mean, you can see overlap with the task and the rest, which is not too surprising, really, because rest is kind of an unconstrained task. But I like that rest is not merely a sum of all of the different tasks that you do. You see a lot of states present in rest that are not there in the task scans, which suggests that there's something that's maybe a little bit different about doing resting state analysis than any of these particular tasks at least. So along with, how different patterns of activity are distributed in this latent space, where I should remind you that bright colors here mean that many time points have been placed in the same area, meaning they're very similar, and so those show up as bright spots. Looking at the same bright spots in the bottom, we can also calculate how long a person tends to spend in any one of those spots. And so high velocity here is shown as a bright yellow color, low velocity is red, and what you notice is people tend to stick in those high amplitude areas that we're really calling states, and they move quickly through areas that don't really have defined states, both for task and rest. So that all makes sense. Now, if we look at these latent space distributions and consider them as kind of like topographical maps, right? So if you imagine that these high density areas that show up in bright yellow in the top row correspond to high mountain peaks, and things that are blue correspond to low valleys, you can use this watershed algorithm to segment different areas from that distribution. And that's what we've done in the middle column. And then you can pick a particular section of that we're calling a state and outline it in pink. And shown next to it at the far right is the wavelet coefficient as a function of frequency there, so the wavelet decomposition for that particular state. And you see that for the state at the top, we have a little bit of lower than average power in some of the low frequencies, whereas for the state at the bottom, we have high power in the very low frequencies and low power in the higher frequencies. So we can see distinct patterns across these states. And I'm just going to kind of go through this quickly. I don't want to take too much time. We can separate this out by task, and you see different patterns for different tasks. We also find that the decomposition is very reproducible across scans when we had multiple runs, for example, first resting state and second resting state. Now, one of the things we found very interesting is that if you look at, time point by time point within a particular task, you can see differences that kind of suggest there might be these trajectories of activation within the brain. So here we're looking at two different components of one of the tasks with random dots, one where the dots move randomly, and one where they kind of move in a coordinated manner, which we've labeled as mentalizing on the top. And we can see slight deviations as the task progresses in the probability distribution for those two types. And if we look at the brain states shown up at the top right, we can see that the main difference between the two is in the very low frequencies in the visual cortex. But this pattern, looking at now the bottom left, this pattern of kind of progression across the latent space gives us a hint that there might be trajectories in the brain. So when we started thinking about trajectories, the first thing that came to mind was that maybe we could use a variational autoencoder. Now, these are network architectures that are widely used to take a signal. You force it through a bottleneck where it's represented by a handful of latent variables, and then you reconstruct those latent variables through a symmetric network to try to recapture the original signal. And the idea is that this gets rid of noise in a lot of situations. So you're trying to denoise a signal using a variational autoencoder. What we were interested in was whether those latent variables in the bottleneck can actually capture fundamental spatiotemporal trajectories that serve as the building block for the resting state fMRI signal. So I'm showing here an early approach where we only used 8 orthogonal latent variables in our reconstruction, so you can ignore the probability distribution on the left. But if you look at the right, now what we've done is encode resting state fMRI from a single individual during a single scan where red is at one end, purple is at the other end, and so you're seeing the temporal progression of that scan through this latent space. And there again are hints of a trajectory, and I think you can kind of see, there's this tendency to go along the diagonals from lower left to upper right, from upper left to lower right, and maybe that's something we can pull out. So we ended up using the autoencoder architecture that I showed as the example, and that we found by some empirical tests that 32 latent variables worked fairly well. And so when we took the Human Connectome project data and fed it through this autoencoder and then looked at our latent variables, we got some things that I think are very interesting. So it's difficult to describe these latent variables. They inherently have dimensions of space by time, and each point has some weighting of these variables. So the way we've chosen to show them is for this particular display, we are showing the spatial components, each parcel from each network is a row, and we're showing its activity over time. Blue is lower than average activity, yellow is higher than average activity, and basically we've gotten these plots by setting each latent variable to its maximum and zeroing out all of the others and then looking at the reconstructed signal. And so if you look at the top row, you see a lot of variability over time. In the second row, there's less. And as you go down, these explain less and less of the overall variability in the signal. What we also found is we looked at the distribution of the spatial variability for each one of these latent variables, and there should be 32 here. And so if you look across space, which means looking at a column, you'll see a black line for each of these. And that shows where we see the most variability across the networks at one time point. And so what we did is cluster these variables by how similar their spatial profiles at that time of maximum variability were. There are a lot of ways to do this. This was just for kind of display purposes and characterization. I should also say that by comparison to other data analysis techniques, we found that the first row looks very much like our primary quasi-periodic pattern, which also has ties to the primary functional connectivity gradient. Second cluster looks like the second functional connectivity gradient. So there are kind of external validations for these. So here's the same thing. Now we are showing for each cluster the spatial distribution as we go from a negative value of that particular latent variable to a positive value. And so you can see the spatial similarity really clearly because we're only showing this now at the time of maximum spatial variability we indicated in the previous slide. We can calculate the functional connectivity within each of these spatiotemporal trajectories, and we've done that along the bottom, so you can get a feel for how different those patterns are. And then in this last one, we're showing the timing of activity in a single ROI that we've picked. And so now you can see along the vertical axis, we have the value of the latent variable, and along the horizontal axis, we have the timing. So you can see, for example, even though this first row has common spatial profiles, we actually have very different temporal behavior for these different latent variables. So we're very excited about these, and this work is still ongoing. I think we have a lot of things we can do to explore how well these characterize resting state fMRI data. But I want to have a little time to talk about another approach that we're using, which is trying to understand the brain as a complex system. And people have been doing this for a long time, and a common tool that they use is a brain network model. And the way you get one of these is you take a graph that describes the network structure of the brain, which we can obtain, for example, from diffusion-weighted fMRI. And at each of the nodes, each of the parcels that we make in the brain, we drive it with a set of differential equations that describes a particular neural mass model, and there are many of these. And from that, we can simulate resting state fMRI data and see how well we recapitulate features that we see in the systems level empirical data. Now, I mentioned that there are many different models you can use, and in fact, one of the problems I think with this approach is that many models and many different parameterizations give the same general average functional connectivity that we see empirically. So we needed a way to try to sensitize our analysis a little bit more. And we did that by adding time-varying metrics. Again, I don't really have time to go into this too much, but I mentioned the QPPs before. So if you look at the top left graph, that's what our QPP looks like from empirical data. And you can see there's kind of this positive correlation and negative correlation and areas switch between the two. Compared to the brain network models, and we've looked at two different ones here, the firing rate at the top right and the Koretsky model at the bottom left, you can see both of them catch this two-state solution, but you don't get this complex propagation between areas that shows up as a sort of smeary pattern in the resting state fMRI data. Now, we did find that the pattern that you get from these brain network models depends on your coupling parameters. And so that's evidence that this dynamic metric is sensitive to the types of parameterizations we use and might help us do a better job. So we thought we needed a new approach. We have dynamic metrics that do a better job, but we still have this huge space of parameters that need to be swept for each model. And so it seems like a great application for machine learning. And we've taken two different approaches for this, and I am going to walk you through those pretty quickly here. So the first one is, instead of using a parameter suite, let's learn the initial conditions. And so for this, we used a network architecture based on a long short-term memory network, an LSTM, which is a recurrent neural net. And we put in a certain amount of the data and fit that to one of these brain network models and tried to use our LSTM to estimate the initial conditions at the end of that block of data. From the end of that block of data, then, we can integrate those differential equations and see if our path follows our data. So here it's demonstrated on a spiral data set where we know exactly which differential equations were used to generate the data. And so you can see we do a pretty good job of fitting it based on those initial points that we learned. Here, we wanted to show that this is also sensitive to the structural network that you use. And so at the top left, we show the fit we get when we use the correct structural network for this particular system. And then at top right and bottom left, we're showing the fits that you get if you use the wrong network. And as you would expect, we have the same initial conditions, so things start out good, but they diverge from the real activity over time if we use the wrong structural network. So they are sensitive to that. Okay, so how does it look when we apply it to resting state data that's empirical? Well, we tried looking at accuracy as a function of the global coupling strength that we built into the model. And what happened was that the accuracy actually went down as the global coupling strength went up. And that's shown on the graph on the left. And so, global coupling strength is usually set to about 0.9 when people do parameter sweeps, and that's our worst performer. So we realized what was happening is that putting any structural constraints on the data actually makes it perform worse. This is supported by the fact that if we use just the LSTM, which is the second point from the right in that graph, we actually do better than we do with any structural network at all. So we had another idea, and we thought, okay, well, let's see how it responds if we mess up the structural network. And so on the right, we're showing the accuracy when we permuted, we've kept only 10% of the original edges of the structural network and permuted the rest, all the way up to keeping 100% of the structural network correct. And you can see if we have a low global coupling parameter, like at the top, we don't really distinguish across these cases. It doesn't matter what we do to the structural network. But if we go to the high coupling parameter, as you see at the bottom right, we start to see this difference as we go towards that correct structural network that we think might help us be more sensitive to which is the right parameterization to use. So we're very excited about how this is coming, and we think this is going to help maybe avoid some of those parameter sweeps. Okay, another thing that we can do, so that last approach, we learned the initial conditions and then we integrated from there. The LSTM was no longer involved. So if you're off on your initial conditions, you're going to slowly diverge from the signal that you're trying to model. So what if we update those initial conditions every time? And so that's what we've done with this approach. Here we do the same thing where we take an initial chunk of the data, we put it through the LSTM, we predict the initial conditions. From there, we feed those initial conditions, we integrate those initial conditions to the next point, feed it back into the model, and then do it all again. And so what we get out of this model is a time course of initial conditions for each time point of the scan. And so this is actually pretty cool. We've done this with two different models, the firing rate model and the Wilson-Cohen model. And you can see if you look at the graph on the left that the estimated and measured resting state MRI are quite similar. You can also see this in the middle. We're showing the time course for each of the different parcels and how well it's predicted based on this LSTM model compared to the original measured signal. But what I think is really exciting about this is now we get estimations of underlying features like excitatory and inhibitory activity, for example, with the Wilson-Cohen model, that we can pull out separately from this resting state MRI data. So I think that will be very cool. So I mentioned before, that dynamic analysis methods really give us the best distinction between different models. And so we looked at the QPPs again, this time from a brain network model that's shown at bottom left and should look similar to before. You see the two-state solution. The empirical data one is shown at the top left. And then we've looked at QPPs and the output from these LSTMs using either the firing rate model for the middle or the Wilson-Cohen model on the right at the top. And what you see is that we're starting to catch some of that more complex propagation from area to area that we couldn't get out of that original brain network model. So we think that this is going to help us fit our data a little more precisely. So both of those projects are still clearly ongoing, but I think I'll stop there with a quick summary. I think machine learning holds huge potential for helping us to get the most out of these enormous resting state MRI data sets. And maybe this is a way to get rid of some of those parameter sweeps. Maybe it's a way to make them more accurate. But most of all, I think that machine learning lets us have a data-driven approach that might help us discover new things, like these latent spatio-temporal trajectories that we might be able to use to explain what's going on in the brain, and that gives us a new way to think about how activity might be organized across the whole brain. Of course, there are always bad things to go along with the good, and I think it's important not to do just a black box application of machine learning, where you may not get something that you can interpret. I'm sure other people will talk about the need to be careful with the data. If you put junk in, you're going to get junk out. And, sometimes I feel like the enthusiasm for machine learning gives us a lot of papers that are published but are very hard to interpret in any meaningful way. Now, I said, we shouldn't use machine learning as a black box. I think we've got to tie it to something that we can interpret. And then that makes it a very powerful tool for us. So some of the things that might be possible are multimodal measurements, maybe behavioral performance. And of course, as in anything in neuroscience, reproducibility is a major issue. And I do think it's important to compare results to existing analysis methods. One of our early iterations of the variational autoencoder was great at giving us principal components, which we could get out of a standard PCA analysis. So, you know, I think there's no need to use a fancy machine learning tool if you have something that's simpler and more interpretable that will work. Now, the last thing I want to show is that, you know, I said we need to tie these things to multimodal experiments. And so we're trying to walk the walk. And what we are doing is setting up simultaneous measurements of optical excitation, inhibition, and then fMRI. And the goal is to take that Wilson-Cohen model that we've used and that we've shown we can learn excitatory and inhibitory currents from and test the activity that we actually measure optically from genetically targeted indicators against what we predict from the resting state fMRI acquired at the same time. Now, we're still working on this. It's technically challenging, but I think it's the right kind of goal to strive for. And I think the mere fact of forcing your machine learning approach to learn something interpretable opens the opportunity for doing this type of validation. So I just want to end with an acknowledgement to my lab, especially Amrit Kashyap, Xiaodi Zhang, who did a lot of the work that was presented in this talk, including, along with them, a former student, Jacob Billings, who is a postdoc now in Czech, and did a lot of work on the t-SNE representation. Thank you very much for listening, and I look forward to our discussion.